Strategy for retrovirus vector-mediated gene delivery. It involves the deletion of essential genes gap, pol, and ENV from retrovirus myeloimmunine leukemia virus, which make it replication deficient. It creates a space for insertion of an expression cassette. The modified retrovirus acts as a vector for the expression cassette. Although replication defective, it is still infectious. Infection of a packaging cell line that carries intact GAG, pol, and ENV genes allows the modified retrovirus to reproduce, and packaged retroviral virus can be collected and used to infect a patient. In the cytosol of the patient cell, the DNA copy of the viral RNA synthesized by viral reverse transcriptase, which accompanies with viral RNA into the cell. This DNA is then randomly integrated into the host cell genome, where its expression leads to production of the expression cassette product. Retroviral genome 5' to 3', vectoral genome 5' to 3', packaging cell viral vector GAG, pol, ENV. The GAG, pol, and ENV genes are detected from the retroviral genome and replaced by the therapeutic gene. The size sequence is retained. This is recognized by viral proteins for assembly of the RNA into a virus particle. GAG, pol, and ENV functions are supplied by the packaging cell, but the genes are on physically separate molecule and when is not a 5 sequence. This is done in order to minimize the risk of producing replication competent viruses. Recombinant viral genome are packaged into infective but replication deficient virus particles which bud off from the cell and are recovered from the supernatant, adapted and modified from human molecular genetics. This picture. Adenovirus. Adenovirus are viruses that carry the genetic material in the form of DNA. When these viruses infect a host cell, they introduce their DNA molecule into the host. The genetic material of the adenovirus is not incorporated into the host cell genetic material. The DNA molecule is left free in the nucleus of the host cell, and the instructions in this extra DNA molecule are transcribed just like any other gene. The only difference is that the extra genes are not replicated when the cell is about to undergo cell division. So the descendants of that cell will not have the extra gene. This means that treatment with adenovirus will require regular doses to add to the missing gene every time. Adeno-associated virus. Adeno-associated viruses, or AAV, from the parvovirus family are small viruses with a genome of single-stranded DNA. There are a few disadvantages to using adeno-associated viruses, mainly the small amount of DNA it can carry and the difficulty in producing it. This type of virus is being used, however, because it is non-pathogenic. Most people carry this harmless virus. In contrast to adenovirus, most people treated with adeno-associated virus will not build an immune response to remove the virus and cells that have been successfully treated with it. Human diseases that are target candidates for somatic cell gene therapy. Hemophilia A, liver, muscle, bone marrow cells, factor 7. Hemophilia B, fibroblasts. Familial hypercholesterolemia, liver, severe combined immunodeficiency, bone marrow cells, T cells, immunoglobulopathies, red blood precursor cells, then cystic fibrosis, lung airway cells, Hershey's disease, bone marrow cells, macrophages, cancer tumor cells. Cloning of mammals. In 1997, Ivan Wilmut announced that the sheep had been cloned from a somatic cell nucleus from an adult female sheep. This was the first time that an adult vertebrate had been successfully cloned from another adult. He took cells from the mammary gland of an adult 6-year-old pregnant Eve and put them into culture. Culture medium was formulated to keep the nuclei in these cells at the resting stage of the cell cycle G0. They then obtained oocytes, the maturing egg cell from the different strain of sheep, and removed their nuclei. The fusion of the donor cell and the enucleated oocyte was accomplished by bringing the two cells together and sending electrical pulses through them. The electrical pulses stabilized the cell membranes, allowing the cells to fuse together. The resulting embryos were eventually transferred into the uteri of pregnant sheep. Of the 434 sheep oocytes originated using in this experiment, only one survived. Dolly, thus it appears that the nuclei of adult somatic cells can be totipotent. But remember this number, 434 sheep oocytes originally used in this experiment, only one survived. So their commitment is commendable. Mammary epithelium disperse grow in culture, induce G0 phase, then nucleus, pipette, ovum, eunucleate, fuse, activate, in vitro embryo culture. In vitro embryo culture, implant, get a new sheep. Cloning sheep by nuclear transfer. The nucleus of an ovum is removed with a pipette. Cells from the mammary epithelium of an adult are grown in culture and the G0 state is induced by inhibiting cell growth. A G0 cell and an eunucleated ovum are fused and the eunucleated form is grown in culture or in ligated oviducts until an early embryonic stage before it is implanted into a foster mother, where development proceeds to term. Human Genome Project, or HGP. The Human Genome Project was a collaborative international government and private sponsored effort to map and sequence the entire human genome. HGP was formulated by the US Department of Energy and NIH. The Human Genome Project's ultimate goal was to discover and map all the approximately 35,000 human genes and make them accessible for further biological study. To facilitate the future interpretation of human gene function, the Human Genome Project also conducted parallel studies on the genetic makeup of other organisms such as yeast, fruit fly, mice, and Arabidopsis. After years of multi-billion-dollar research, the Human Genome Project and Celera Genomics, a non-government biotechnology company, jointly initiated drafts of the human genome sequence in 2000. By mid-2001, scientists associated with these ventures had presented the true nature and complexity of the digital code inherent in DNA. We now understand that there are approximately 35,000 genes in human nuclei DNA, comprised of approximately 3 billion chemical bases arranged in precise sequence. Salient features of human genome. The human genome contains 3164.7 million nucleotide bases. The average gene consists of 300 base pairs. Gene codes for protein dystrophin consist of 2.4 million base pairs. The total number of genes estimated is about 35,000. The size of chromosomes in between about 55 MB and 250 MB. Chromosome 1 has maximum number of genes and Y has the fewest. About 98.5% genome is one coding, only about 1.5% of the protein coding sequence. Repetitive sequences make up very large portion of the human genome. Highly repetitive sequences comprises less than 10% and transposable elements about 50%.